Oh, hello, students. We will just wait for maybe 2 or 3 more minutes and see if anyone else joins, and then we can start with the session. Okay? And I also hope that the screen is visible. Okay, so we will start with the with today's session. Let me introduce myself to you. And I am Prashasti Pandey, a PMRA Fellow and PhD Scholar in Banaras Hindu University, Department of Botany. And I'll be taking today's discussion session of the course Plant Developmental Biology, whose course instructor is Professor Shri Ram Yadav from IIT Roorkee. And so far we have conducted three such sessions, and this is the last session where we will be discussing few of the questions of previous year's assignments, hoping that these questions and their answers will be helping you in your current ongoing assignments and will be of use during your examination for this particular course. And okay, so with this, we'll just start with the session. I'll be showing some questions on the screen of previous year's assignments. And if you know the answer, please go ahead and tell the answer. If you don't know it, and if nobody answers, I'll tell the answer and a brief explanation to that question. And if there is any other doubt, any other question that you want to ask or discuss something, we can do that towards the end of the session. Okay, so we'll just proceed. Here is the first question. Which of the statements is false regarding reverse genetics? First is targeted suppression of the gene by its down regulation, making a null mutant of gene of interest, mapping of mutant gene of interest, and none of the above. I'll give you 2 minutes. If anybody knows the answer, please tell it. Otherwise, I'll give the answer and the explanation to it. Okay, so before telling the answer, we'll just proceed with understanding what reverse genetics is. Okay. So basically, we have, if any one of you has attended the previous sessions also, there was a question based on the similar topic, forward and reverse genetics. And let's try to understand this again. There are two types of basically studies in which, while we are studying genetics, one of them could be forward genetics and the other one is reverse genetics. Now, how do we confirm or how do we say that this particular study is a forward genetic study and that this is a reverse genetic study? So suppose, for example, if we are talking about plants, and if we have a plant with certain trait or any kind of phenotype or physical feature, and based on that physical feature, we go towards the direction of identifying the gene which is responsible for that physical feature, then the study from phenotype to the genotype will be called as forward genetics. Similarly, the reverse genetics will be the study from the genotype to the phenotype, where we already know that this particular gene is responsible for this phenotype. We are just trying to, either mutate the gene or alter that gene in any form, and then try to see that what kind of phenotype it is generating. So when we are doing this kind of study, it will be called as reverse genetics. Okay, so I hope this forward genetics and reverse genetics concept is clear. And if the concept is clear, you can obviously understand that any technique which is involved in identifying of gene, like sequencing techniques like gene mapping or map-based cloning or BSA or shore map or something like these kind of techniques where you basically try to identify the gene from the phenotype will come under the techniques used for studying forward genetics. And any kind of technique which will be used to alter the phenotype, which will be like silencing of the gene or deleting the gene by CRISPR-Cas technology, or silencing the effect by RNA interference, or using tDNA or insertion or removal of transposons. So techniques like this will actually alter the genotype and then we can study the phenotype that has been generated from the alteration of that particular genotype. And therefore, these techniques will be used to study the reverse genetics approach. Now, with this information, coming back to our question, which is which of the statements is false regarding the reverse genetics. First one is targeted suppression of the gene by its down regulation. So yes, targeted suppression of the gene. So if we targeted suppression is nothing but using the RNAi technique or the CRISPR-Cas9 technique, and these techniques will be used to alter the gene and thereby altering its phenotype, which will be later studied. So yes, this is true about reverse genetics. The second one is making a null mutant of gene of interest. So here also, if you try to understand the statement, we are talking about mutant of gene of interest. That means we already know the gene of interest. We just don't know that what will happen once we remove that gene. That essentially translates to trying to understand the phenotype that is the outcome of mutating the gene of interest that we already know. So of course this will be true in case of reverse genetics. The third one is mapping of mutated gene of interest. Mapping of mutated gene of interest will actually come under the forward genetics, because the gene has already been mutated and we know the outcome. We are just trying to map of where the gene of interest is lying, or where, so basically we are trying to study the genotype here and not the phenotype. And if we are studying the genotype, that means that will come under forward genetics. So mapping of mutated gene of interest is not related to reverse genetics, and therefore the answer to this question is mapping of mutated gene of interest. I hope this question was clear, and we'll just move ahead to our next question. Which of the following correctly represents the receptor, inhibitor, response promoter of auxin and its downstream signaling? Your options are DR5, AUXIA and YUC, TIR, YUC and DR5, TIR, AUXIA and DR5, and none of the above. I'll give you 2 minutes. If you know the answer, please go ahead. Okay, so let's just to answer this question first understand auxin signaling. Okay, so I am sure that you people would have studied auxin signaling, but we'll try to understand it here. That what happens in case of auxin signaling. So suppose um, here is the auxin targeted gene. Okay, so auxin target gene is nothing but any gene which will be transcribed as a response to auxin which is generated in plants. Okay, so all the plants have a promoter region, and these promoter regions have certain elements which are called as cis acting elements, or they are basically very small 8 to 10 nucleotide base pair sequences to which some of the either hormones or some metals or some ions, these kind of things bind, and they help that particular gene to transcribe. So we have similarly we have this OXRE, which is present in the promoter region of the OX targeted gene. So for the OX target gene to transcribe, this OXRE needs to be activated. If OXRE is not activated, the transcription will not happen. So how can this OXRE be activated? So this can only be activated when the ARF, which is bound to this OXRE, will activate this OXRE. So basically, this is supposed to activate OXRE, and then OXRE will transcribe the auxin targeted gene. But how this ARF is supposed to activate OXRE when it is bound by OXIA? Okay, so now this happens when there is no auxin present. Okay, first situation is that auxin is actually absent in our plant, and in that case, the auxin will this particular machinery is active and the transcription is not happening. Now let's consider a situation where the auxin is present in our plant. So now what is this auxin will do is this will recruit this TIR1 AFB factor to bind to this OXIAA. So this is the auxin molecule. Okay, so only in presence of this auxin molecule, this OXIAA will bind to the TIR1 factor, and once this entire complex is formed, by the method of ubiquitination, this entire process will be degraded. This entire complex will be degraded. So now what happens because of this degradation is this OXIA, which is stopping this ARF from activating OXRE, gets removed. Okay, so this is not present, and now because this is not present, this ARF will get activated, and this ARF will in turn activate the OXRE, and this OXRE will help the transcription of auxin targeted genes, and this is how the auxin targeted or auxin signaling actually happens. In this case the transcription will happen. Okay, so I hope I'm clear with this thing, that what we are trying to say here is that in presence of auxin in plants, auxin will help binding of OXIA1 and OXIA and TIR1, and this entire complex will be degraded by the ubiquitination method. And once this is degraded, the ARF will become free, and ARF will activate the auxin responsive elements present in the promoter region of the auxin target gene. And once this auxin responsive elements get activated, the promoter will help the transcription of its target gene, and that's how the signaling will the signaling will happen. Now the question is that what is this OXRE? So this is a small element which is also called as DR5, whose sequence is TGT-CTC. Okay, so when this sequence is present in any promoter region of any gene, that means it can bind to the ARF factor, which means it is regulated by the presence of auxin. Okay, so any gene which has ARE element or DR5 in its promoter region can be regulated by auxin. And this is one example. So if you can see here, this is a GFP gene. GFP gene is nothing, if you have attended the previous discussion sessions, GFP is a kind of a reporter gene, and it is attached with this auxin regulated gene here. So once this entire machinery, that is auxin, is present, auxin will help binding this OX with the TIR1, and this ARF will get activated. It will bind to ARE and help in the transcription of this GFP gene, which is why you can see the green fluorescent color over here. Okay, so this is the auxin signaling. Now coming back to our question, which of the following correctly represents the receptor? So receptor here of auxin was TIR1. So basically the answer to this question is the answer to receptor is TIR. Okay, now inhibitor. Inhibitor, basically the inhibitor here, as we know, if you go back and try to understand from here, is OXIAA. Why? Because when this OXIAA is bound to this ARF factor, the transcription cannot happen. It will only happen once this OXIAA will be removed. Therefore it is called as the inhibitor. So here the answer to this inhibitor is OXIAA. Okay, and the third part is response promoter. So response promoter is DR5, which was the sequence to our auxin responsive element. Okay, So now the answer to this question will be TIR, OXIA and DR5. So this will be a correct answer. I hope the question was clear. And now we'll move ahead with our another question. So the next question is, during establishment of leaf polarity, the portion which is towards the meristem is dash and the portion which is away from the meristem is dash respectively. The options are proximal distal, abaxial adaxial, distal proximal and adaxial and abaxial. You have 2 minutes. Please read the question thoroughly. Okay, so to understand this question, let's just understand the leaf polarity first. So there are two surfaces of the leaf. One of the surfaces is called as adaxial surface and the other one is called as abaxial surface. So now when you take a leaf, the top side of the leaf, which has the trichomes and which has this the palisade mesophyles present just below the trichomes, and the surface that is actually exposed to the sunlight is called as the adaxial surface. The surface which is on the below side, which is not exposed to the sunlight as much, and basically which is not directly exposed to the sunlight, and there are more spongy mesophils present, and also there are most number of stomata present on this particular side will be called as the abaxial side. So the lower surface of the leaf will be called as the abaxial side, and the upper surface of the leaf will be called as the adaxial side. Now, since the question was during the establishment of leaf polarity, so that means the stage will be when the leaf is formed. So now try to understand from this figure. See, if you try to see this B figure first, this is the leaf of a plant called as Arabidopsis, which is a model or organism belonging to the Brassicaceae family, which is a model plant basically. So if you see at the leaf of this particular plant, you can see very tiny these structures present here. This white colored structures, small spike-like structures. So these are called as trichomes. And if you see the other surface of the leaf, very few, almost no trichomes are present. And these the ones that are visible are because of the folding of the leaf from the sides. They are also present on the other side. So there are almost no trichomes present on this side. So this side will be called as the abaxial side and this side will be called as the adaxial side. Also, there is other terminology called as distal and proximal. So these two words are used with respect to their presence either towards the stem of the plant or away from the stem of the plant. So the proximal side will mean that this will be present. So if suppose this is the stem of the plant and from here the leaf is growing, so this surface will be called as the proximal surface, and the surface, the part that is away from the stem will be called as the distal surface. Okay. So now coming back to our during the development of the leaf. So here is the mesophyll tissue. Okay. Now from this mesophyll tissue, the leaf will start to grow. Okay. So just imagine that when the leaf will start to grow like this, then there will be one surface, this surface here, which will be towards the mesophyll cells, and there will be this surface which is away from the mesophyll. Okay, so now when the leaf will, so suppose it will start developing like this and then it will expand and then form a leaf here. Okay, so basically this surface which was towards the mesophyll will come on the top side, and the surface which was away from the mesophyll will come on the bottom side, and the part which is attached to this mesophyll will be called as proximal side, and the part which is here basically away will be called as the distal side. Okay, so once the leaf starts growing, so the part which is near to the mesophyll will develop into the above surface of the leaf, which is called as the adaxial surface. And the part which is away from the mesophylls will develop as a lower part of the leaf, and therefore it will be called as the abaxial surface. Now, coming back to the question, during establishment of leaf polarity, the portion which is towards the meristem. So now this portion will be towards the meristem. So the portion which will be towards the meristem will develop into the adaxial side, and the portion which is away from the meristem, that is this portion, which will develop as a lower part of the leaf. This will be called as the abaxial side. So the answer to this question will be fourth option, which is adaxial and then abaxial side. The one which will be away from the meristem will be the abaxial side, and the one which will be towards the meristem will be called as the adaxial side. And that's how the polarity of the leaf is decided during its development or during its establishment. Okay, so I hope this question was clear. We'll move ahead to the next question. Okay, so I'll just read out the question loud. In the Fabulosa mutants, organization of vasculature is, the options are xylem outside, phloem in center, phloem outside, xylem in center, xylem adaxial and phloem abaxial, and phloem adaxial or xylem abaxial. Uh, Please read this question and let me know if you know the answer. Okay, so to understand this question, let's just first understand what fabulosa mutants are and then come towards its organization of the vasculature. So basically, fabulosa mutants are the plants with dominant mutations in the PHB transcription factor. Fabulosa is the full form of the PHB, and these mutations basically are responsible for altering. So basically, PHB transcription factor gene is responsible for leaves polarity, and if the gene is mutated, then the mutations will be caused in the polarity of the leaf, which will basically cause the transformation of the lower side of the leaf to become the upper side and the upper side to become the lower side. Also, with these mutations, there are certain root developmental defects also. So, once these kind of mutations happen in any plant, the leaves, so there are three major impacts that PHB mutations will cause. The first one is that in the leaves, the PHP mutants will basically transform the lower side of the leaf into the upper side. And in the roots, these mutations will have defects in the root meristem and its growth. And also, it will cause basically supernumerary cortex formation during early root development. So these are the, because of these things, there will be developmental impairments in the root as well. Fabulosa mutants basically, so when we are saying that the leaf polarity will change, that means, so vasculature is nothing but the arrangement of the xylem and the phloem. Now, in normally the case, what happens is that basically the xylem is often, it is surrounded. Okay, so the xylem and the phloem, they are present in a concentric circular form. Okay, so basically xylem is inside and the phloem tissue is present outside. But because of the mutation in the PHP gene, the reverse arrangement of the vasculature happens where the xylem completely surrounds the phloem. And this essentially reverses the pattern where xylem gets located inside the vascular bundle and the phloem comes outside. So this happens because of the fabulosa gene, because it plays a very critical role in the arrangement, in causing the polarity, in arrangement of the tissues, of the vascular developmental tissues, and its movement towards outside and inside. So when these movements and these developments are impaired, the exact opposite happens of what happens in the normal plant. And here the xylem basically, so xylem gets located inside the phloem and phloem comes outside. Now, fabulosa is a class 3 HZ-CYP transcription factor which helps regulate development of vascular tissues, and particularly determining the xylem and the phloem arrangement. So that is why when the fabulosa gene is mutated in any plant, the arrangement gets disrupted. Now, if you see here in this figure, the this is again an Arabidopsis plant. If you try to see this figure, the leaf here is inverted. Okay, the lower surface becomes on the upper side and the upper surface gets inverted in some form and the leaf becomes this kind of disrupted. Okay, so these are the Arabidopsis mutants of this particular gene. Okay, I'm sorry, I'll just. Okay, so that this is the basically the Arabidopsis mutant of the Fabulosa genes. And now coming back to the question, in Fabulosa mutants, organization of vasculature. Vasculature is the organization of the xylem and the phloem. So the answer to this question is that xylem comes outside and the phloem comes in the center. This is the vasculature organization in the Fabulosa mutants. So this is our next question. Please read the question and see if you can answer it. Okay, so I'll just read the question. Members of which gene family typically express in every floral world and gives floral organs its identity? The options are A class, B class, E class, and C class. Please let me know if you know the answer, or else we'll proceed with understanding this. Okay, so now let's try to understand this. And this particular thing of understanding the floral world and the gene responsible for development of the floral organs is called as the ABC model of flower development. Now, let's try to understand this ABC model. So basically, we have three genes, and these three genes have their particular gene names. But to make it easy to understand, let's just name them as A, B, and C gene. Okay, so now if you arrange these genes in this particular manner, so this represents that this particular part is basically when both the B and the C genes will express together, and this overlapping region will be called as when B and A genes will express together, then certain a phenotype will be generated, and the non-overlapping regions will be the ones where only, like this region, so this region will be the one where only the one single gene is generated, and what happens with the with that phenotype. Now let's try to understand this ABC model. So what we understand from this figure is when only C gene is expressed, then only the gynoecium is formed, or the carpels are formed. Okay, so when the C gene expresses, the gynoecium part of the flower is formed. When B and C genes together express, then androecium of the flower is formed. Now, when A and B genes together express, that is when the petals or the corolla is formed, and when only A gene expresses, that forms the sepals and the calyx. So now, by missing or removing of any of these genes, these organs which are developed because of their expression will either be formed or be removed from the floral arrangement. There are two more basically genes which expresses in this ABC model. One, the other ones are called as one is D gene and one is E gene. Okay. So this is a paper from this is a figure from a paper from Nature Reviews, and we'll try to understand this ABC model with this figure. So so basically the D gene that I talked about. So D gene expresses only in case of ovule formation. Okay, so when the ovule has to be developed in any flower, that is when the D gene will express. So now that means that it will not play a role in the whorl formation of the flower. So whorl will consist of calyx, that is sepals, petals, androecium, and gynoecium. So when we are saying that flower development in terms of whorl development, then these four things will come into play, and there are four genes which are responsible for that: A, B, C, and E gene. Okay, D gene will be responsible for the development of ovules. Now when you see this A figure here, okay, so if you see A figure, we are saying that all the four genes are getting expressed here. That is A, B, C, and D, E gene is also getting expressed here. That means all the four organs will be formed by either their individual expressions or their co-expressions. So if you see here, we have here the sepals, we have the petals here, and here we have the androecium and the gynoecium. Now let's come to this B figure. In B figure, we are removing the expression of the A gene. So here there is no A gene expressed. Now if you look back at the figure, we are saying that by removing only A genes, the sepals get removed. So here if you see, if you understand this arrangement of the flower, this is called as a floral diagram. So if you see this, the only one part of the flower is removed here. Okay, so we are saying that A gene is removed here. So this A genes will remove the sepals. But this B gene, when because there is no A gene, the A and B expression, because they are getting expressed together, B alone gene cannot produce the petals as well. So in this case, the petals will also get removed. The petals will not be produced in this case. Okay, so if you see in this figure, there are no petals in this case, because petals will only be produced by the co-expression of this A and the B genes together. Okay, so now if you see the first C figure, okay, so in the C figure, we are removing the B gene. Okay, that means the phenotype which will require the expression of A and B or B or C genes will not happen in this case. So here only sepals and calyx will be formed. If you go back to this figure, if you remove this B genes, that means there will be no androecium and there will be no petals. So only sepals and gynoecium will be present in this flower. So if you see this flower, here in this case we don't have a C gene. So in this case there will be no androecium and there will be no petals. Okay, because we are removing the B gene. So because of this B gene, there will be two things that were getting co-expressed in presence of A and C genes, which was androecium and petals. So both these things will get removed. Now here we are removing the C gene. Sorry, here we had removed the B gene and we are removing the C gene here. Okay, so once we are removing the C genes, that means our A gene is getting completely expressed. Our gene with A and B co-expressing together is also happening. But the C part and the co-expression of B and C will not happen in this case. So if you see this figure, if you remove the if you remove the C part from here, that means the gynoecium and the stamens will not form. That means the flower will be infertile. Okay, there will be no reproductive organ in this particular flower. It will have the petals, it will have the sepals also, but there will be no androecium or no gynoecium, because the C gene is not getting expressed. C alone will be expressing the gynoecium part, and C in co-expression with B part will be expressing the androecium. So there will be no reproductive organ in this particular flower. Now if you see the E flower, So here none of the genes are expressing A, B, C, and the E gene is also not expressing. So such plants will produce a leafy rosette like the cabbage flower or the cabbage. Basically cabbage is nothing but a leafy rosette flower because it will have neither it will have the sepals nor it will have the petals, neither it will have androgen nor it will have gynosion, because none of the world producing genes are getting expressed in that flower, and therefore the flower arrangement will be nothing but a bunch of leaves arranged which look like a flower. So this is called as the ABC model of flower arrangement. And now if we come back to the question, which of the gene family typically expresses in every floral world and gives floral organ its identity? If you go back to the figure and look carefully, we have this gene with class E expressing everywhere, okay, in all type of arrangement. So no matter what the flower arrangement is, this E gene will always express. So the answer to this question is E class. I hope the question was clear. Let's move on to the next question. Okay, this was the last question. So if you have any doubts, please let me know. Otherwise, we can end this session. Okay, so if there are no questions, we can end this session. And if you need any another discussion session, please raise a request for that. We can conduct another session if it is required. If not, all the very best for your exams. And yeah, if there's any other doubt, just let me know, or we can end this.